When the job is finished, the results will be available in the Land tab under Analytics, Open Results Set. In Sample Grouping to Expression, you will find the results in table format, with the list of genes that were found to be significantly different, along with the results from the Kruskal-Wallis analysis, including p-value and adjusted p-value. Gene expression for each group for the no or adenocarcinoma, as well as yes squamous cell carcinoma, will be shown here, along with minimal and maximal expression of the gene in the entire sample set. This view can easily be sorted, such as by p-value, by right-clicking on the column header and choosing Sort. Genes can also be filtered in the View controller, for example, filtering for a maximal expression of at least 3 to filter out genes that have a low expression. To visualize this data in Land, users can search for the top 100 or so genes by clicking on the top gene and scrolling down to find about 100 genes. While these genes are highlighted, using the keyboard to copy, Control-C, we can then go to the search bar at the top of the screen, click the drop-down, and search multiple genes. By using Control-V, we can paste these genes into this as a list and search these genes within Land. This search will search all available data in the Land related to these genes. This may take 20 to 30 seconds to search depending on the speed of the connection to Array server. The default view, when this is done, will be the DNA Alteration View, where users can see how frequently these genes are affected in different tumor types. Let's go back and filter to our samples of interest again, this time using the saved metadata columns. For tumor type, let's first right-click on tumor type and choose the checkbox instead of radio. This will create a checkbox filter in which we can select multiple tumors in the analysis a little later on. For now, let's choose esophageal cancers only, or ESCA, by right-clicking and saying check this only. Then, under sample type, let's choose primary tumors only. Finally, let's choose samples with RNA-seq transcript data. Once we've filtered to the samples of interest, let's see how these genes are expressed by Select View, RNA-seq Quantification, HeatMap View. In this view, hierarchical clustering is performed showing these genes on the y-axis and individual samples on the x-axis. It is clear that there are two main clusters of samples based on the expression of these genes. One on the left with the lower expression of the majority of these genes, and another on the right with the higher level of expression of these genes. To see how this clustering relates to our grouping, we can add back color bars under the Task, Change X-axis color bars, and add a color bar related to our sample set. Under Sample Set, Squamous, we can slide this to the listed columns, and our heat map is instantly updated. Under Legend, we can see that green refers to squamous carcinoma samples, and we can see that the squamous carcinomas cluster very well with the samples that have low expression of these genes, whereas the adenocarcinomas mostly cluster with the samples with high expression of these genes. This view is highly customizable. For example, by right-clicking on the legend, change color, we can change, for example, the middle color from white to black. As we slide this over, we see that our view is instantly updated. We can also adjust the brightness shown within our heat map.